QuorumBio is a bio-venture company with a research center in Seoul National University. We are developing super antibiotics that suppress resistant super bacteria and Alzheimer's treatments using Quorum Sensing inhibitors. Biofilm protect bacteria from substances such as antibiotics and can be said to be a defense fortress of bacteria that exchanges resistant materials. The biofilm is not created simply when bacteria gather, but must go through a process called Quorum Sensing. You can see it's connected by a very sticky substance, like a barrier. In order to be made like this biofilm, it must go through a process called Quorum Sensing. Bacteria control not only biofilm but also group behavior through Quorum Sensing. When bacteria recognize that a certain number has gathered, bacteria begins expression of a new gene. Quorum Sensing induces not only biofilm, but also group actions such as antibiotic resistance and virulence expression. As such, Quorum Sensing is a necessary process for bacteria to develop antibiotic resistance. Biofilm, the starting point of the group action of bacteria, increases antibiotic resistance and plays an important role in the acquisition and release of resistant materials. Inside the biofilm, antibiotics cannot function properly because biofilms are created only when there is Quorum Sensing of bacteria. QSI inhibit biofilms and increase the antibiotic effect. In addition, recent research shows that when used with antibiotics, it can destroy already formed biofilms. We are developing new concept drug using QSI. Currently in the preclinical stage, we want to license out. Pseudomonas aeruginosa and Acinetobacter baumannii are bacteria that are detected in large amounts in pneumonia and sepsis. In particular, Acinetobacter baumannii is a major causative agent of nosocomial infections in hospitalized patients with impaired immunity, and the development of a new treatment is urgently needed. As a result of quantitative analysis of the biofilm, it is inhibited greatly. Our candidates inhibited biofilm formation of E. coli, one of the bacteria that can cause various infectious diseases, such as urinary tract infection, pneumonia, and sepsis by acquiring resistance to carbapenem class antibiotics. In addition, since effective biofilm suppression ability was confirmed for chronic periodontal disease, bacteria and major gram-negative bacteria, such as F. nucleatum, P. dingivalis, T. and forsythie, it is expected that the scope of application will be extended to several strains that have not been tested. Our candidates have the ability to suppress the expression of virulence factors. In addition to inhibit biofilm, it was confirmed that it inhibits the release of proteolytic enzymes, which are major virulence factors, and also suppresses the expression of FADA, a bacterial host cell adhesion factor. This result proves that not only biofilm inhibition, but also virulence can be inhibited through Quorum Sensing inhibition. This is the result of confirming the effectiveness of candidate in animal models. The first is the result of analyzing the level of inflammatory cytokines induced when infected with bacteria, while the expression of TNF and IL-8 is significantly increased by bacterial infection. The level of inflammatory cytokines is decreased during treatment with the candidates. The second result shows that, in the periodontitis mouse model, alveolar bone resorption was decreased in the group administered with the Quorum Sensing inhibitor. The third result confirms that the inflammatory activity of the brain immune cells, astrocyte and microglia, is increased by bacterial infection in the rat model of brain inflammation, whereas the inflammation of the brain tissue is relieved when the QSI is administered. This data is the result of evaluating the antibiotic efficacy enhancing effect of candidates. In the MIC test for Acinetobacter baumannii, Pseudomonas aeruginosa, and Escherichia coli, the main causes of multidrug-resistant bacteria, It was confirmed that the efficacy of the existing antibiotic was increased by treatment with the Quorum Sensing inhibitor. It can be seen that the efficacy of several antibiotics, such as ampicillin, cefepime, meropenem, and amenetem, was significantly increased. In an animal experiment to evaluate the safety of the drug, no adverse reactions were observed in the skin, tissue, and body for 1 week after administration of the candidate substance. We evaluated the pharmacological and pharmacokinetic actions of the drug to prepare it for preclinical trial of QAL 333. Plasma stability in humans and animals was very good, and it can be confirmed that it is slowly decomposed in the evaluation of metabolic stability. As a result of CYP inhibition, to evaluate drug-E interaction, there was no problem in co-use with other drugs. Also, no specific problems were found in the cardiotoxicity evaluation. This is the result of a pharmacokinetic evaluation. To determine the route of drug administration, pharmacokinetic evaluation was conducted through various routes of oral, intravenous, and subcutaneous administration. In the case of subcutaneous administration, it was predicted that it would remain in the body for a long time with one administration. In addition, since it is not necessary to consider the problem of disturbance of the intestinal microflora and hepatotoxicity, which must be considered during oral administration, preclinical studies are conducted with subcutaneous administration. In addition, in the blood-brain barrier BBB penetration test that we've additionally tested, it was confirmed that the candidate material penetrates the BBB well, so it can be an effective drug even when septic bacteria penetrate the brain. QuorumBio is developing a treatment for atopic dermatitis by creating healthy intestinal microbiota. This substance, which has a Quorum Sensing inhibitor function, has excellent suppression of atopic dermatitis and is very safe because it is an edible natural product. PreGalactose has a probiotics function that provides nutrients for beneficial bacteria, such as mucin-degrading bacteria in the intestinal mucosa, and a function of weakening adhesion and virulence by inhibiting Quorum Sensing of harmful bacteria in the intestine. It is safer than probiotics, and the stability of the substance is very high enough to be boiled. We have obtained patent for atopy and obesity for this substance. We have proven that PreGalactose is highly effective in improving atopy. When PreGalactose was administered to mice with severe atopy, most of the atopic dermatitis were cured. The effect was almost identical to that of mice administered with the immunosuppressant dexamethasone. Positive control. We conducted metagenome analysis of intestinal microbiota to study how intestinal bacteria changed in atopic dermatitis animal experiments. Mucin-degrading bacteria Akkermansia, Mucinophila, and Rhaminococcus navis are known to cause problems with immune function in the human body when their colonies are reduced in the intestine, which leads to atopy. When PreGalactose was administered, MDB significantly increased. The ratio of Prevotella and Lactobacillus species, which are known to alleviate clinical symptoms of atopic dermatitis, also increased. We have already reported in papers that PreGalactose effectively inhibits Fusobacterium nucleatum, the bacterium that releases the most A2, and also directly inhibits AI2 action. Due to the AI2 inhibitory effect, it was confirmed that the pharmacutes over bacteroidetes ratio, which was broken when DNCB was treated, was restored to normal when PreGalactose was administered. PreGalactose grows mucin-degrading bacteria such as Akkermansia and Luminococcus, and these bacteria protect the mucin layer and immune cells. In addition, by inducing the formation of a healthy and normal microbiome, it improves atopic symptoms by blocking the production and penetration of inflammatory materials. PreGalactose also inhibits the action of the Quorum Sensing factor A2, preventing colony formation, adhesion, and invasion of harmful bacteria. By maintaining the balance of bacteriotics over firmicutes, it forms a healthy microbiome and prevents the onset of atopic dermatitis. Thank you for your attention.